Hey, it's Mr. Shrum. Welcome back to a biology walkthrough. And today we're going to look at genetic mutations. And the objective here is to make and defend a claim based on evidence that inheritable genetic variations may result from genetic mutations. So for a warm-up, we are asked, is the following statement true or false? Genetic mutations are always harmful? Not necessarily. They don't always have to have harmful effects on organisms. They can just be neutral, and some may even be beneficial for the organism. So let's move on to the next warm-up question. The white wallaby in this image has a mutation that gives it a white coloring. How could this coloring affect its survival in its environment? Well, it would negatively affect their survival. A brown wallaby can camouflage itself. It can blend in more easily with the surroundings. But this white wallaby would easily be seen by predators and stand out as a sore thumb. And that can't positively affect their survival. So to start off, we're going to look at gene regulation. Protein synthesis occurs in the cell because of two processes, transcription and translation. Gene regulation is the ability to control which genes are transcribed at any given time. And this gene regulation happens in both prokaryotes and eukaryotes. And if you remember, prokaryotes are unicellular organisms that lack a nucleus, while eukaryotes are a unicellular or multicellular organism that has cells with a defined nucleus. But it happens a little differently in each of those cases. Here's a chromosome, and then if you zoom in, we see that it contains DNA for gene A and gene B, which is what we'll use as examples. So first, we're going to look at prokaryote gene regulation. This is carried out with the help of operons. And an operon is a section of DNA containing the genes required for transcribing a particular protein. The operon mechanism allows prokaryotes to produce proteins only when required, often in response to environmental cues. An operon consists of these three parts. We have a promoter, where the enzyme Rna polymerase first binds to the DNA, the operator, which is like the on-off switch. Structural genes are codes for specific proteins. So we have the promoter, operator, structural genes. A special regulator gene plays a role in the operon mechanism by coding for proteins that control operon expression. However, because the transcription of the regulator gene is not under the control of the promoter and operator of the operon, it's not considered part of the operon. Let's look at two operons found in the bacteria E. coli. Trp operon. Along with the promoter and operator, the Trp operon of E. coli consists of 5 structural genes. And these 5 genes contain the genetic code required to synthesize enzymes that are essential for the biosynthesis of the amino acid tryptophan in the bacterial cell. The regulatory gene for this operon is TRPR, and it's located somewhere in the bacterial chromosome. It codes the TRP repressor protein, and with the help of the TRP repressor protein, the TRP operon controls the synthesis of the amino acid tryptophan in response to its availability in the environment. And you can see that happening in this diagram. The regulatory gene, the promoter, here we have the repressor, and then this is considered the TRP operon. We have 1, 2, 3, 4, 5 structural genes. When tryptophan is available in the environment, it's taken up by the bacteria. Within the cell, tryptophan binds to the inactive TRP repressor protein and then activates it. So this repressor was inactive, but then the tryptophan binds to it and activates it. And once it's activated, this complex of repressor protein and tryptophan then binds to the operator of the TRP operon and blocks it. Now RNA polymerase cannot bind to the promoter to begin transcription. Therefore, the TRP operon is repressed. If there is no tryptophan in the environment, the bacteria will need to code the structural genes of the TRP operon. In absence of the tryptophan molecule, the repressor protein will not be activated and will not bind to the operator. Now the RNA polymerase can bind to the promoter and initiate the transcription and expression of the TRP operon. Since the operon is turned off in the presence of tryptophan, it is called a repressible operon. So if an operon can be turned off in the presence of something, it's a repressible operon. And here in this diagram, we see the repressor, the tryptophan. Tryptophan binds to the repressor, tryptophan repressor complex. Complex means once these two are combined, they are called a complex together. And them, the repressor and the tryptophan, once bound together, they bind to the operator, and that means no more transcription of TRP operon. LAC operon, this operon contains genes that allow bacteria to break down lactose. So Lac Operon, lactose, and that is a milk sugar. This operon is induced or activated when glucose is not available and lactose is the only available source of sugar. The Lac operon consists of a promoter and operator and 3 structural genes. The structural gene code for enzymes needed to break down lactose. So these genes code, this genetic code allows enzymes to break down lactose. And the regulatory gene, LACL, or LACL, encodes the LAC repressor protein. And that is located adjacent to the Lac Operon. When glucose is available as a sugar source, the Lac repressor is active. It binds to the operator in the Lac Operon and does not allow transcription of structural genes. So you're noticing a pattern here. When something's present in the environment, one thing happens, and when it is not present in the environment, another operation happens. When glucose is absent and lactose is present, the Lac repressor gets inactivated. The inactivation of the Lac repressor protein induces or activates the Lac Operon. So when one's activated, the other one is inactivated. The molecule allolactose causes the Lac repressor to become inactive. And this allolactose is an isomer. And that means it's structurally different version of lactose. And when allolactose binds to the Lac repressor, it changes the shape of the Lac repressor. And as a result, the Lac repressor can no longer bind to the operator. This allows the RNA polymerase to bind to the promoter and carry out the transcription of the structural genes. Because the Lac Operon is turned on in the presence of the inducer allolactose, it is called an inducible operon. So we have repressible operon, and then we have inducible operon. So when something can be turned off in the presence, since the operon is turned off in the presence of tryptophan, that is called a repressible operon. And then because the Lac operon is turned on in the presence of the inducer allolactose, it is called an inducible operon. So checkpoint. Imagine you are introducing the Lac operon and the TRP operon to students who have never learned about it before. Complete the table to compare the similarities and differences between the two operons. And here we go, we have the answers. So the TRP operon is a repressible operon. It's deactivated when tryptophan is present in the environment. The repressor protein, it's activated by tryptophan. Transcription, tryptophan binds to the repressor protein, activating it. So notice this trend with repressible operon. It's deactivated when something is present in the environment. The repressor protein is activated by something. In this case, it's tryptophan. And then tryptophan binds to the repressor protein, and that activates it. Now, Lac operon is an inducible operon. It's activated when lactose is present in the environment. And the repressor protein is inactivated by lactose. And then the transcription, lactose binds to the repressor protein, deactivating it. So repressible deactivates when something is in the environment. The repressor protein is activated by something in the environment. And then tryptophan or something binds to the repressor protein, and that activates the repressor protein in repressible operons. And then Lac operon's inducible, something, it activates it when it's in the environment. And that same thing inactivates the repressor protein, and then that something binds to the repressor protein, deactivating it. So remember this table because it can be helpful distinguishing those operons. That was prokaryote gene regulation, and now this is eukaryote. Gene regulation in eukaryotes is more complex because several steps are involved in gene expression. Eukaryotic gene regulation involves the interaction of many genes that are controlled by more than one promoter and one regulator. Most genes are regulated at the level of transcription through transcription factors. These factors are proteins that control whether a gene is transcribed and the rate of its transcription. They ensure a gene is made at the right time and that the right amount of protein is made. There are 4 main types of gene regulation mechanisms in eukaryotes. Regulation of transcription, that's DNA to messenger RNA or mRNA. Regulation of processing of pre-messenger RNA, that's also known as splicing. Regulation of degree of messenger RNA degradation, that's known as messenger RNA stability, and regulation of translation of messenger RNA, which is control of protein synthesis. And we have a video on transcription and translation for an overview of the process in eukaryotes. So here we go.
You really are what you eat. The protein you eat is broken down into amino acids. Then it's absorbed and used by your cells to build the proteins that your body needs to survive. Scientists estimate that you have around 37 trillion cells in your body, and most of them are microscopic protein-building factories. This image might look like a mess, but it's a picture of a protein factory. It's pretty gross, huh? But it's super, super tiny. There are several of them in just one cell. No factory can operate without workers and blueprints. So you can imagine that protein blueprints and protein workers are even smaller. Let's take a look. Your cells have a set of plans written into the code of your DNA, and your cells use these plans to build the proteins that become new cells and tissues. Protein synthesis has 2 major steps: transcription and translation. The goal of transcription is to make an RNA strand that the ribosome can use to build a protein. Transcription starts with your DNA inside the nucleus. The DNA molecules are unzipped, and an RNA strand is produced with the help of an enzyme called Rna Polymerase. DNA is made up of nucleobases called adenine, thymine, guanine, and cytosine. When DNA is transcribed into RNA, the thymine bases are replaced with uracil. When this RNA strand is assembled, it's called messenger RNA. Messenger RNA, or mRNA, is like a delivery service. It leaves the nucleus and brings the message to the ribosome so the correct protein is made. But before it leaves the nucleus, messenger RNA needs to be edited. The messenger RNA made from DNA contains introns, which are non-coding regions, and exons, which are regions that code for proteins. The introns are spliced out, and the edited messenger RNA leaves the nucleus and travels through the cytoplasm to a ribosome. This brings us to the second major step of protein synthesis. Once the messenger RNA is in the ribosome, the process of translation begins. Translation is the process in which the strand of mRNA is read by the ribosome and proteins are built. If you think of the ribosome as a factory, think of the mRNA as a blueprint or a template. It's the map that the ribosome will follow to create new proteins. Here's how the process of translation works. A series of 3 base pairs on a strand of mRNA is called a codon. A codon can contain any combination of adenine, cytosine, guanine, and uracil. Each codon codes for a specific amino acid. Another type of RNA molecule called transfer RNA, or tRNA, acts as construction workers in a ribosome. Each transfer RNA has a specific amino acid attached, and it matches up to a specific codon on the mRNA strand. Take our AGU codon from earlier. Adenine bonds with uracil. Guanine bonds with cytosine. So the tRNA that's carrying a UCA codon will bond to an AGU codon on the mRNA, just like 2 building blocks or perhaps the tumblers of a lock and the teeth of a key. As the transfer RNA matches up to the codons on the messenger RNA, the amino acids on the transfer RNA form peptide bonds, forming a protein chain. This protein can then be used by the cell or sent outside of the cell to another part of the body. Your body is constantly building proteins, such as rebuilding muscle fibers, growing hair and nails, and creating gross stuff like snot and sweat, so you can do all the things you want to do.
Nice. So that was a pretty good video overview of what transcription is all about. Let's look at some of the mechanisms of gene regulation. Regulation of transcription. Hox genes are an example of gene regulation at the transcription level. Hox genes are a group of related genes that regulate the formation of various body parts. They are active at the embryonic stage of development. Hox genes code for transcription factors. Transcription factors determine which genes are expressed and their quantity. The transcription of a Hox gene therefore determines the body plan of an organism. Hox genes are found in clusters and could be spread over different chromosomes. Humans have 39 Hox genes located in 4 clusters on 4 chromosomes. The fruit fly, Drosophila melanogaster, has 8 Hox genes. They are clustered into 2 complexes, ANT-C and BXC, Antenopedia complex and the bithorax complex. Regulation of messenger RNA degradation and translation. The mechanism of RNA interference, RNAi, regulates the degree of degradation of messenger RNA. RNA usually exists as a single-stranded chain of nucleotides, but it can form double-stranded complexes. Some viruses have double-stranded RNA as their genetic material. Double-stranded RNA, or dsRNA, molecules control the RNAi mechanism through these 3 main steps. An enzyme dicer cuts the double-stranded RNA molecules into small interfering RNA, siRNA, which bind to a special protein. The protein degrades one strand of the siRNA and forms a ribonuclease or RNase complex with the other strand. The ribonuclease is an enzyme that degrades RNA, and this complex binds to a specific messenger RNA and degrades it, preventing its translation. Geneticists are exploiting the RNAi mechanism by introducing synthetic double-stranded RNA molecules to silence targeted genes. This mechanism has the potential to find methods to treat cancer, diabetes, and other diseases. All right. Question. In labs, scientists have introduced mutations in fruit fly genomes to create flies with legs growing where their antennae should be. Explain why changing the nucleotides in the Hox genes could cause this. Well, Hox genes control how body parts are shaped and arranged. Genes are sections of DNA that code for a particular protein. They give the organism its visible traits. And if you alter the nucleotide sequence in this section of DNA, that alters protein synthesis. This change causes a completely different body part to grow. Interesting. And I think I will stop there for now, and we'll finish this next time. But until then, I will see you all later, and have a great day.